We're going to start with the tutorials by Sam Isaacson from Boston University. So this will continue in the vein that Dave Anderson prepared us for stochastic simulation for models without spatial components. Now Sam will discuss how we're going to include the spatial components.
Okay, thanks. Yeah, so I should say, stochastic simulation of spatial models is an enormous thing, and I could tell you about everything from cell movement to fluid dynamics inside cells to tumor growth and so on. I'm going to kind of pick, I think, the natural extension of what Dave talked about, which are kind of stochastic chemical kinetics models where we add in spatial diffusion and maybe tomorrow, if we have time, some other transport mechanisms. So kind of the extension of the well-mixed models from yesterday to the case that things are moving spatially. And I should say, my plan here is that the first lecture is going to kind of be an overview, so I'll kind of go through everything that I'm hoping to touch on, maybe even more than what I'll get to over the next 3 lectures afterwards, where I'll then zoom in on specific details of how we formulate things mathematically and derive some of the simulation algorithms. And of course, it gives you an opportunity that if this is of no interest to you after the first lecture, you can just bail out and you don't have to go to the next 3, and you'll still have an overview of what I'm going to talk about. Okay, so there's many different scales that when we're thinking about modeling processes inside cells we can look at. And of course, my ideal world, we would write down a Hamiltonian for our cell and solve the Schrodinger equation and be done. But as we all know, there's no way we can do that. And so we heard yesterday talks actually across many different scales, mainly at this end. So Dave told us a lot about stochastic chemical kinetics. Linda told us a bit about the models using the reaction diffusion master equation, which I'll mention in this first talk and probably go into more detail about tomorrow. And of course, then there's interesting questions about how these are all linking, how you can go from the kind of particle-based stochastic reaction diffusion models I'll discuss today to, say, well-mixed stochastic chemical kinetics models or deterministic reaction diffusion PDEs, and then vice versa back to the mass action ODEs. And when I made this slide, which I stole from a talk I gave several years ago, this was kind of the state of the art for how large a system you might simulate with these types of, whoops, well, that's way ahead, okay. You might simulate with these types of systems. But actually now there's groups that have gotten up to simulating order 1 million proteins, maybe even a little more. But it's a lot of work using, say, GPUized codes where you're splitting across multiple GPUs and you're doing kind of all kinds of fancy tricks to speed things up. But that kind of gives you a feel for the scale that probably the largest scales that you can conceivably get to. But the person who'd be a real expert if you want to know how big a system can I simulate with these models is probably to bug Linda. Okay, so kind of the outline for my first talk here is I'm going to tell you a little bit about why I'm interested in stochasticity in chemical reactions, and in particularly why I'm interested in incorporating explicit spatial movement of proteins in mRNAs. Then we'll go through kind of the types of particle-based stochastic reaction diffusion models that people have used to study problems in cell biology. So there's basically kind of 3 models or simulation approaches people take. So we'll go through the kind of formulation of the models and then talk a bit about how you simulate them and some trade-offs in the different simulation approaches. And then at the end, I just want to show you kind of one example of this, of what's kind of state-of-the-art for what people can do. And actually, it was state-of-the-art 3 years ago, so my guess is they can simulate even bigger systems now. Okay, so maybe I don't need to, in this audience, convince you that stochasticity in biochemical reactions is important, but still let me kind of mention it. And of course, the classical example, right, is evolution. If every time your cell divided, that was a completely deterministic process, evolution could never occur. Your daughter cells would be genetically identical to the mother cells. So stochasticity is key in that process. In the context of kind of what I'm going to talk about today, a better example maybe is gene expression and regulation. Right? So if we have a very simple negative feedback autoregulatory system, gene makes mRNA, makes protein, the protein can dimerize, and the dimer can inhibit and shut off the gene. What we see is if I look at the steady state distribution of the number of proteins in a population of cells, that's what each of these histograms shows, the steady state protein distribution across the population. And if we vary the rate that the dimer comes off the DNA, so here the rate is very large, so it only stays bound a short time. Here the rate's very small and it stays bound a very long time. We see that when the rate is sufficiently small, we get these kind of bimodal distributions where parts of the populations are completely turned off and no transcription and no protein is available in some of the cells. And this is very much an effect of the stochastic model. You don't see kind of bistability in the corresponding deterministic model. This is only something that emerges in the stochastic model. Now, today I'm talking about spatial movement in cells, and probably everyone in the audience knows about this, but it's worth mentioning. So the primary way that I'm going to think about things moving in cells is by diffusion. Of course, that occurs both in the cytosol nucleus. In the classic example which I think David Holkman will tell us about, is maybe the search for DNA binding sites in the next tutorial. Very often we have diffusion to things that are membrane or scaffolding bound, and I don't know if I'll have a chance to talk about modeling that at all. But that's, of course, a huge problem where there's still a lot of work to be done. And one of the things that can be challenging is there can be a very broad range of diffusion constants, from very slow diffusivity of mRNAs to very fast diffusivity of, say, small tracer molecules that we're injecting into cells. And I just want to show you that you can look at these things experimentally. So what you see here, and this is very old, so you can do even better nowadays, are traces of individual mRNAs moving inside the nucleus of a Chinese hamster ovary cell. So this is the nucleus. In orange, you can see, I believe that's a histone label, so think of that as the regions of heterochromatin where the densest DNA is. And here you can see paths traced out of individual mRNAs, and on sufficiently short kind of space and time scales, the paths look diffusive. But there may be kind of effects where they can't get into these regions of very high density. But if you kind of analyze them when they're moving in these open green regions, you see that it looks diffusive. Okay, and of course, the other way that things move around is active transport, which I'm going to pretty much ignore probably in my talks, but I'm happy to chat more about it. And of course, this is mainly used out in the cytosol. And we can think of this, right, as rapid directive transport. It's kind of the railroad system of the cells. We've got here, in this case, microtubules, and this is individual kinesins moving along them. And you can see very clearly they kind of move in this rapid motion along the tubule that they're attached to. Yeah?
Can I ask something? Just
Yeah, sure. And I should say, ask questions as much as you want. Yeah,
just
that's
to have
the
a
point.
sense of scale. So you say that it's both diffusion and transport. So in terms of scale, is the transport happening in a smaller scale? So is this sort of like, have you zoomed even more in the cell?
So I don't know the length scale of either of these movies. I'm going to guess that this is, order 10 microns. This one I have no idea. I'd have to look it up. This is something I put. But active transport is going to take you generally over much longer length scales because it's fast directed motion. So if you want to move from a nucleus out along, say, a dendrite to far away from the nucleus, active transport is a much better mechanism to get your cargo out there.
I'm just asking from the naive point of view. For example, when you in that image, for example, you only see diffusion. Does it mean that if you were to look closer, you would see transport? That's basically
This image. This one.
You see diffusion.
Oh, here I don't think there's any transport going on here because it's in the nucleus, and as far as I'm aware, there's not really active transport of mRNAs in the nucleus.
So that would be outside the nucleus.
This is out in the cytosol. In fact, this may be in vitro. It may just be something that they've set up kind of in the lab where they just have microtubules and kinesins that move on them. So,
Thank you.
Yep. Okay, so like I said, today and most of my tutorial, I'm going to focus on diffusion. Okay, so why care about this spatial movement? Can we just account for everything with non-spatial models? Why actually try to take this into account? Well, there's been some examples that have shown that diffusion can be very important and play a key role. And I don't have time to go into a lot of details of these, so I'd suggest maybe looking up some of these references. In particular, there's a very nice paper by Neves et al. From Cell that shows that when you have neurons, both the size and shape of your neuron can have a significant control on the dynamics of negative regulators out in dendrites. And in particular, that modulates these kind of microdomains of activated signaling molecules. So basically, out in the periphery of the dendrite, the kind of diameter of the dendrite, which is what changes here, can have a big effect on the kind of local spatial gradients and therefore the kind of signaling that you see in the dendrite. And that can affect this downstream pathway. This is an example of, I think, cyclic AMP signaling pathway that they looked at. Even if you ignore geometry and you take a perfectly kind of uniform, well-mixed, just cube, Johan Elf showed quite a while ago at this point that if you have slow diffusion, that you get something that may look very different from a well-mixed system. So here's an example where he showed that in this kind of, I think it's a double negative feedback system, you can kind of have these kind of segregation into islands where regions go to kind of one chemical state and other patches go to another, just from having slow diffusion. Whereas if you looked at the kind of total population, you wouldn't see any segregation. You might see basically that the total population of red and blue is the same. Okay? So here, things very much don't look well mixed spatially, yet it wouldn't be clear that there's any kind of separation going on if you just looked at kind of non-spatial statistics. And of course, both these examples that I've showed you, they're completely ignoring the fact that inside the cell is not really a spatially homogeneous test tube, right? The inside of a cell is very complicated. And that's one of the things that I'm very interested in. So I thought, since I'm giving a tutorial, I can at least show you a couple slides on things I work on. So this is an example just to show you what the inside of a eukaryotic cell looks like. I'm going to show you X-ray CT image of a human B cell. So the cell's been frozen and placed in a glass tube. And the movie I'm going to show you is just moving along the z-axis through the cell. And the nice thing here is that the pixel intensity in the movie that I show you will be directly proportional to the density of organic material. So basically where you see white, except for the boundary, there'll be very high density of organic material. And where you see black, there's very little organic material. So it's very low density. And so we'll start moving through our cell. So here's the cell now. And here's the nucleus of the cell. And you can see a lot of structure here. You see sheets of ER. You see mitochondria in white. In the nucleus, you can see regions of heterochromatin in light and euchromatin in dark. You can see actually the Golgi in here. And what the movie shows, I think, very clearly, right, is that the cell very much is a spatially heterogeneous environment. Okay? So at least to me, I'm kind of interested in what role that heterogeneity might have in cellular processes. This is just a reconstruction showing the organelles. It was a segmentation of the organelles from that same data set done by Marc Lagrosse. So I'm going to move now through the cell. And that we won't show the ER in the first one, but we'll show the others. So here's the nucleus. This is mitochondria. In the nucleus, you can see euchromatin and heterochromatin. This is the Golgi apparatus. And now we're going to come back through the cell, adding in the ER. And as you come back through, you really see, so much of the space is occupied by the organelles, at least in this particular cell. It really fills quite a lot of space. Okay. So there's a lot of spatial heterogeneity, and it's kind of interesting to me to think about how that might come into play in some of these chemical pathways that we study. So I wanted to show you one example before we kind of get on to the meat of the tutorial, just to illustrate that. And I'm happy to talk about this more if it's of interest to people. So what we've done here is we've looked at simulations of the mean first passage time it takes a protein starting at a given point on the cell membrane, so on the surface of the cell, to first reach the nuclear membrane. Okay, so it's just diffusing through the cell. And we've looked at two sets of simulations. One where all those organelles are removed, so the cytosol is just an empty region, so there's no obstacles. And one where those organelles are all present. So here you see the nucleus in gold, that's the target you're searching for. You see the mitochondria in green, you can see the ER. So in the second simulation, all those organelles are present and they represent excluded volumes. So basically, you reflect off them. You can't move through them. And the question is just how does the physical presence of those organelles as obstacles affect the time to go from cell membrane to nuclear membrane? And so what I'm plotting here is the ratio of the time when you have organelles as obstacles to the time when you have an empty cytosol. And what you'll see is that it's always at least about a factor of 2. That's dark blue. These red regions, it's actually as much as a factor of 10. And it's interesting to kind of think about where you have the largest regions, but that's something we can talk about after. And one thing to point out here, the ER in this movie, which shows you where the organelles are, I have to kind of attenuate it so you can see in. Otherwise, you wouldn't be able to see into the cell. Okay. But we can see quite a lot of variation. So if a signal hits the cell here, it's going to get to the nucleus much quicker than, well, I should be a little careful, actually, because this is showing the relative change. So the relative change is much bigger in those regions than, say, in these regions. The absolute values looks a little different.
Yeah, this is the mean first passage time to any position.
To go from a specific point on the cell surface to any point on the nuclear membrane. I'm assuming essentially that pores would be, uniformly distributed, and it's basically, you can think of it as time to get to a pore if you have a lot of pores.
Did you look indeed at the mean first passage time to a pore?
I did not look at the mean first passage time to a specific pore, but we could certainly do that. I mean, I don't know if that's a reasonable thing to look at, though, because usually, right, I don't think you're going to be localized to one specific pore, right?
But
to any pore.
Yeah, because they only represent
right. So I didn't.
2%.
Right. So this is just getting to the nuclear membrane surface. then getting to the pore and through the surface would be another step on top of this. That's right. So that would be an interesting thing to do, but we haven't looked at that.
Sorry, dumb question. So is it faster when you have the organelles?
Oh, no, sorry. Yeah, sorry if I wasn't clear. It's slower. So this ratio is always at least about 2. In fact, you'll see on the histogram on the next slide. So maybe this makes it clear. So this is the distribution of times at points on the cell membrane surface. And what you see here is the distribution when you have no organelles. So the mean first passage time distribution across the surface. And then in blue is when you have organelles. So you see you have a much sharper, more narrow distribution, and it flattens out and widens quite a bit. So in general, the times get much slower. And so basically, the presence of organelles acts as an obstacle, a barrier that slows you
More
down.
density, they could help.
Yeah, so it's interesting to try to think about when could it speed you up or slow you down. But we've looked at 6 cells, and for the 6 cells that we've done, doing these segmentations takes a week and a half. We found in every case that there's a pretty substantial slowdown. So, okay, so I could give a whole talk on these kind of simulations, and that's not the point of his today, but I just wanted to point out this example, and in particular, this is just for one molecule. What's going to happen if we try to look at a whole chemical pathway, like MAP kinase or something in these types of geometries? But that's a question for the future. Okay, so that was just a little motivation on why I'm interested in these types of problems. So now, we'll shift gears a little bit, and I'll tell you about the types of stochastic reaction diffusion models we're interested in using to study these types of problems, and give you an overview of those, and then we'll come back to talking about how you might actually simulate them. Okay, so I say, what are the 3 stochastic reaction diffusion models? That's a little ridiculous. There's probably billions of stochastic reaction diffusion models. So what I mean, these are the 3 that I've seen used quite a bit in modeling in cell biology. So that's how I'm going to think of them. The first one I'm going to talk about, I'm going to call the contact reactivity model. Some of you may have seen it called the Smolchowski model. There's 2 versions of it. One is often called Smolchowski. The other is sometimes called Collins-Kimball. The original version goes back to Smolchowski, and basically now we think of our molecules as moving around in continuous space by Brownian motion, so every molecule just diffuses around. And then reactions happen when molecules reach a fixed separation. So in the classical Smolchowski, or what I'll call a pure absorption model, as soon as 2 molecules reach what's called the reaction radius, RB, they react instantly. Okay? And mathematically, as we'll see in the kind of underlying PDEs for the densities of the positions and numbers of molecules, this corresponds to a Dirichlet boundary condition. Okay? The Collins-Kimball or partial absorption model says that when they hit that separation, there's some chance that they reflect instead of react. Okay? So it just slightly generalizes. That will give you a Robin boundary condition. I'm not going to go too much into that case in the tutorial, though it is used in many simulations. Okay. So that's model 1 at a high level. Like I said, we'll go into a lot more detail of the mathematical description of these models in the next lecture. Here I just want to kind of go at a high level, and we will see a little bit of the equations in a few slides. The second model we'll talk about is what I'll call the volume reactivity model. It's also called the Doi model or the lambda rho model in the literature. And this model is almost the same as Smolchowski. The only real difference is that for bimolecular reactions, now you think of your molecules more as point particles in regards to bimolecular reactions. And so now when they're within a reaction radius, they have some rate of reacting. Okay? So instead of reacting upon collision, which gives us a boundary condition, they react with some rate when they're close enough, which in our PDEs that we write down later on will give us a sync term. Okay? That's the only real difference. And then the final model, which I'll talk about, which we heard in Linda's talk yesterday, is the reaction diffusion master equation. So this one is a little different, and then in the reaction diffusion master equation, space is discretized into a lattice, and we only keep track typically of the number of molecules of each species at each lattice site. We typically assume molecules are well mixed, that is uniformly distributed within their lattice site. So we don't know their exact position, we just know they're kind of uniformly distributed in their box. And then molecules move by continuous time random walk. So every molecule has some probability per unit time. It can hop from its box to each neighbor, okay? Which we'll see is basically we can think of this as like a first order reaction, and so we can use the whole kind of SSA Gillespie method that Dave talked about to simulate these systems. bimolecular reactions will occur for two molecules that are at the same lattice site. They'll have some probability per unit time that they can react to form a complex. Okay? So basically, it's like we take the well-mixed system we heard about yesterday and put it in every box. And then we allow molecules to hop with some rate from box I to box J, which is like another first-order reaction. Okay? So conceptually, it's really kind of very similar to what we saw yesterday. Okay. So those are the three kind of basic models. I just wanted to kind of be a bit more explicit about the differences. And again, we'll be going into much more detail. But in the contact reactivity model, where things react upon collision, then the state of the system we're going to think of as the number of each chemical species and the positions of the molecules. And then the probability densities you're in a given state are going to give us a coupled, usually infinite system of integral partial differential equations with reactive boundary conditions. And by reactive boundary conditions, that's really for bimolecular reactions. 0 and first order, we'll see, just give us terms in the PDE. The volume reactivity model is almost the same, except we replace these reactive boundary conditions with reactive interaction functions in the PDEs. That's the only real change. Okay? And then the RDME, well, now in the RDME, usually we think of the state of the system, like I said, as the number of each chemical species at each lattice site or in each voxel. But the purposes of this tutorial, it makes it easier to compare to these two models if I write it in a similar way. So we'll look at instead the total number of each species and the lattice position of each molecule, which is a completely mathematically equivalent way. It's kind of like Eulerian versus Lagrangian description. And then what we get is that the probabilities you're in a given state is going to satisfy a coupled, possibly infinite system of ODEs, okay, which is just the reaction diffusion master equation. So that's kind of the high-level description. So let's go into a little more detail of what these models look like. And again, if this goes over your head, feel free to ask questions. I'm going to go into this in much more detail as we kind of derive the models and then the underlying numerical simulation algorithms in the next lecture. Okay? So let's look at a kind of simple example. And the reason I pick a bimolecular reaction is that's where things really start to disagree or be a little different between the models. So we'll look at A plus B goes to C. I'll let A of t be my stochastic process for the number of species A in the system. Little a will be a value of it, so A of t equals A gives me the value of the stochastic process. Then we need a way to describe the positions of the molecules, so we'll label them in the following way. Q sub L super A will give me the location of the lth molecule of species A when there's little a of them total. So the superscript tells me the total number of them. The subscript refers to a specific molecule. For now, we'll think in 3D, but we can formulate this in n dimensions. And then we don't want to keep writing all the individual subscripts, so we'll group those into just a species A state vector that has the positions of the A molecules. Okay? So that's our basic notation. I hope that's clear to everyone, because I'm going to be using this notation a lot in the next lecture in particular. This is one of the annoying things about these models, is that unlike what we saw yesterday, I have no idea how to write down a nice process description of these models for general chemical reactions. So in fact, I'd say if someone in the audience knows how to do that, please teach me. I would love to learn that. So we're going to have to write down and study these models through equations for the probability densities we're in a given state. That I know how to write down. Okay, so that's what we'll write down equations for. So here, this is the notation for that. F A, B, C of this will be the probability density that when I have A molecules of species A, they're at the positions in the QA vector, and so on for the B and the C molecules. And there's one technical point which we'll see. This really just comes up in how we normalize things. We want to assume molecules of the same species are indistinguishable, right? Any two A molecules we want to treat the same. So what I'm going to assume is that this is symmetric. So if I take Q sub J A and Q sub I A, the positions of two molecules, and I flip them in the QA state vector, it shouldn't change. It should be invariant. Okay? So we'll assume that this is a symmetric function in the components of these vectors individually. Okay, so what do the underlying evolution equations look like? And we'll go through this on a slightly more detailed example in the next lecture. So we're going to get a PDE of this basic form. Here, L is going to be my diffusion operator, R is going to be my reaction operator, and for every possible number of A, B, and C molecules, I'll get a different PDE, and I'm going to get a big coupled system. So if I had no reaction, my molecules would just diffuse. So here, these are just Laplacians in the QA coordinate, the QB coordinate, the QC coordinate. So it says no reaction, every molecule on its own will just move by Brownian motion. Okay? The reaction operator is a little ugly. So if I'm thinking about the contact reactivity model, so the Stochastic model, then it's going to have to give me the flux coming from the state where I have one more A, one more B, and one less C into the state A, B, C. That's how I would move by a reaction. This would be the state that brings me into this state by a reaction. And I'm not going to write it down now. Maybe we'll write it down in the next lecture, but it's a bit ugly. How would I leave the state A, B, C? Well, this is my reactive boundary condition. It says that when an A and B are separated by a reaction radius, I have an instantaneous reaction, which is just a Dirichlet boundary condition. Okay? So that's the kind of basic model. What this is then going to end up giving you for a general system is, like I said, a coupled system of partial integral differential equations that you're going to get. Okay. Yeah?
Just to clarify, the integral comes from the reaction, yeah?
R.
R.
That operator I'm not writing down, but I will write it down for the volume reactivity model where it's a little easier to write down on the very next slide. So, okay. So this is the last, I think the last bit of notation for these kind of models. If you get this notation down, then you'll be in good shape in the next lecture. So the volume reactivity model, we said molecules react with some probability per unit time when they're closer than a reaction radius. So let's call that probability per unit time for A plus B goes to C lambda. Okay? So this is the rate of reaction when two molecules are close enough. If we're going to have a reaction, we need a way to add and remove molecules. So QA union Q means to the species A state vector, I add an A molecule at Q. QC slash QLC means I remove the Lth molecule of species C from the QC state vector. Okay? Told you the notation can be a little cumbersome. So now you get rid of that reactive boundary condition, and instead you get the following reaction operator in the volume reactivity model. So this looks absolutely horrific, but conceptually it's not too bad. It just says, how do I leave the state where I have A molecules of species A at the positions in QA and so on? Well, if I have an A molecule and a B molecule, this is an indicator function of the interval 0 RB. So if this is 1 when their separation is less than RB, then they'll react with rate lambda, and that takes me out of that state. And then I have to sum over all possible pairs of A and B molecules. How do I get into that state? Well, I have the state where I have one more A and one more B and one less C, and my A and B are positioned to produce the C that I throw away, right? I throw away one C, and then I say I should have an A and B position so that if they react, they give me back that C. And here, I'm assuming, and we'll change this up a little later, that when an A and a B react, the C gets just placed at their midpoint. These kind of little technical things come up all over the place. Where should you put a molecule when a reaction occurs? Where should you put molecules when things unbind, and so on? So here, let's just assume that I put it at the midpoint. Okay, so I hope that that's clear to everyone, because this is kind of the basics of the mathematics. We'll be seeing more of it in the next lecture, but this is kind of the starting point. And then the last model, like I said, is the RDME, which we'll talk a lot more about tomorrow. So basically, in the RDME, our idea is going to be, let's just discretize R3 into a Cartesian grid. So now my molecules live at grid points. So this will be the center of a grid voxel. I'm going to use a standard kind of Cartesian grid of cubes of width h. Now we have a discrete space probability density to be in a given state. And it satisfies an equation of the following form with a discretized diffusion and discretized reaction operator. In the RDME, the diffusion operator is exactly kind of what you might do when you discretize. That is, you replace your continuous Laplacians with the standard discrete Laplacian. And if you kind of write this out, this just corresponds to molecules hopping around with rates d over h squared. Those are the kind of connectivities that you get in your discrete Laplacians. So that's nice. This would converge back to our continuous models, for the diffusion at least. What about the reaction term? Well, the reaction term looks a lot like the volume reactivity model. This is very similar. The difference is that now, instead of two molecules reacting when they're within a fixed separation, they react when they're within the same space voxel. So that is, this is a discrete delta function. If they're at the same lattice site, they would react with some rate k over h cubed, where k is your normal well-mixed bimolecular reaction rate that you would use. Okay? And then here, this is the same kind of incoming term where you have one more A, one more B, one less C. And the A and B's that you add are positioned at where the C is you threw away, so that when they react, they produce a C there. Okay. So this is a little those of you that are familiar with RDME, this might look a little less familiar to you, but you can show that this is completely equivalent to the standard form where you track the number of molecules at each lattice site. for each species. It's just the kind of simple scaling to convert between the two. So you have to convert from density, and then you have this kind of scaling factor that pops up in front. Okay, so that's the kind of basic mathematical formulation of the models. We'll see more detail on that in the next tutorial. I wanted to just mention a little bit, for those that are familiar with reaction diffusion PDEs, how these might be related to those types of models. And the RDME is where I've seen this studied the most, the connection there. Though everything I'm going to show is very much formal, and I would say it's actually this kind of connection between the two is to some degree still an open problem, how to kind of show rigorously the transition between them. So let's suppose that I let A of I be the stochastic process for the concentration of species A in the ith voxel in the RDME. Then you can show with a little bit of work that the average concentration in the ith voxel satisfies the following equation. And if you look at this for a second, you say, okay, this is just the discrete Laplacian right here. So if there's no reaction, if I take my mesh spacing to 0, the average concentration just satisfies the diffusion equation, which is what we would want. Now we have this bimolecular reaction term, and we might say, this looks almost like what we would expect to get in the reaction diffusion PDE for A plus B goes to C. But there's one difference, of course. Here we have the average of AI times BI instead of the product of the averages, right? And generally, of course, it's not going to be true that those two are equal. So if that was true, then we would essentially just have a discretization of the reaction diffusion PDE as the means of the RDME, but that's not true. But let's for a moment suppose it is true and think a little bit about when that might occur. And where we would see that potentially this kind of result might come up or might become true is in certain limits. For example, the thermodynamic limit, where maybe we take large populations like Dave, or large volume limits like Dave talked about. Here I said with H fixed, but actually I think most of the limited results on this I've seen, they have to take H to 0 while you scale up populations too. So there's kind of some limits that come up. L. Arnold has a couple papers on this, and I can give people references if it's of interest. In any case, let's kind of ignore this technical point. Let's assume that we're looking at a limit where we take the lattice to 0, and this decouples, that is the average of the product gives me the product of the averages. Then I can define an average concentration in the limit as H goes to 0, and we'll just get out the standard reaction diffusion PDEs. Okay. So this is very much a formal argument, but the basic idea is that the reaction diffusion master equation, its means modulo bimolecular reactions, really are just discretizations of reaction diffusion PDEs. As long as you have linear reactions, it'll converge back. If you have bimolecular, you have to worry about this issue that the average of a product is not product of averages, and then you have to start considering limits where maybe that decoupling occurs. And like I said, I think it's still very much an open problem to actually kind of go through and figure out in what limits one can make these kind of approximations. I haven't seen a ton of work on it. Okay, good. So let's talk a little bit about how we can actually numerically simulate these models, and in particular what some trade-offs in the different simulation approaches are. And then I'll just end kind of the first part here by showing you one example of what one of these simulations looks like, since I think it's a good idea to have a feeling for what these simulations actually look like at the end of the day. So like I said, there's three kind of models we talked about. The first two were these spatially continuous models, the contact reactivity and volume reactivity models. And there's two main simulation approaches that people have used in modeling cellular systems here. One is Brownian dynamics. And there's a lot of different variants on Brownian dynamics, and there's more than I can list here. I'm going to focus for the contact reactivity model on the Brownian dynamics model that's used by Smolden, which luckily we have Steve Andrews here who can tell me all the ways I get it wrong as I describe it. So I've asked him a million questions this week, so hopefully I've straightened myself out on all the details of how it works. And the basic idea here is they're based on kind of an operator splitting of the underlying PDE to separate diffusion and reaction from each other. And then the operator splitting here is over the course of one time step. So these are time step based methods. Generally you evolve diffusion, evolve reaction over a time step, and then rinse and repeat. And of course that's a very crude way to describe a very complicated method, but that's kind of the high level approach of it. And I should say that Roddick, and I think Kostas has worked on this, and there's probably other people in the audience who I'm forgetting, have worked on a version of this for the volume reactivity model. They originally called it the lambda rho model. And I'm going to kind of describe this simultaneously to the Smolden approach because in some ways maybe it's a little easier not having to worry about boundary conditions, just having these interaction functions instead. Now, this is a time-step-based method, so there's a numerical error. There's actually an exact method for the contact reactivity model that you can use called the first passage kinetic Monte Carlo method. It's also now, there's a method called EGFRD, and this is a little confusing because there was something called GFRD, which was not an exact algorithm. It had kind of a slight assumption, a numerical assumption built into it. And then the first passage kinetic Monte Carlo method came out and kind of showed how you could do things exactly. And then GFRD got updated to GFRD, which gave you the kind of complete method for doing the contact reactivity model with partial absorption exactly. So this is a very nice way to actually do exact simulations of the underlying stochastic process. And you might say, well, why would you ever use an approximate method if you have exact methods? Well, one, these methods are really, really tricky to code up and implement. There's a lot of kind of algorithmic optimization that has to go into them. And they're very tough to extend if you want to have, say, spatially varying drift or diffusivities or many other things. So they only work in kind of certain contexts, but in those contexts, they work very well. And we'll go over the kind of basics of how that works in a few minutes. And then the final kind of approach is the RDME, like we mentioned. That's the final kind of model. And then for the RDME, life's very easy. We have stochastic simulation methods like Dave told us about. We've got Gillespie method, first reaction method, next reaction method, partial propensity methods, every method you can think of. You know, there's 50,000 papers on RDME simulation methods and on stochastic simulation methods. All of them can go over to the RDME. One popular one, which Linda mentioned, is the next subvolume method by Elf and al. And if there's time, I'll try to go a bit over these methods to talk about their different algorithmic complexities at the end of my lectures, since that's something we haven't seen much about so far. And one nice thing here is there's lots of public simulators. We saw PyURDME by Linda's group and Andreas Hellender and others. One that's been taken to very large scales and has kind of GPUized versions is Lattice Microbes, which is a time step based one, so it's not exact, but that allows them to do certain GPU things. And there's a number of other ones. And of course, the nice thing with these simulation processes is that it gives you exact realizations of the underlying stochastic process, at least in terms of the statistics, as Dave mentioned yesterday. Okay, so let's talk a little bit about how these different methods work at a high level. So first, I'm going to talk about the Brownian dynamics, which we said this is a time step-based approach, and it's kind of the basic approach we would use for both that can be used for both contact and volume reactivity models. I'm going to talk here about the contact reactivity model as it's done in Smolden, or my understanding. And I'm just going to think about a simple bimolecular reaction. If we have time, we'll talk in more detail later on about how you could extend it to other reactions or how it works for other types of reactions. So the basic idea is I have my molecules in some bounded domain. The first thing we would do during one time step is we diffuse all our molecules by just sampling from a Gaussian, so let's ignore the domain geometry. Okay? So pretend there's no boundary initially. So that gives us an exact sample of the diffusion process for each molecule. So here you just update the position as follows, where these Cassis are just samples from normal distributions with mean 0 and variance 1. Okay? So it's standard kind of solving the, sampling a Brownian motion. So that moves my molecules. How would I do a bimolecular reaction? Well, if I have two molecules that end up closer than a reaction radius, if I'm doing the contact reactivity model where they react upon collision, then they should have reacted, right? If they're overlapping, they had to collide. And therefore, at the end of my diffusion step, I check. And any two molecules that overlap, I just say, OK, they've reacted, okay, instantly. If you're doing the partial absorption case where you have a probability of reflecting, things are a little trickier. And I'm not going to go into that here. So that's the basics. I feel like something disappeared there. I guess not. OK, so that's kind of the basics of how you do it ignoring the geometry, kind of at a very high level. There's lots of things I'm leaving out. So I didn't tell you about how you would do first or zero order reactions, but you can imagine that each molecule has its own clock that gives it an exponentially distributed time that it would change an A molecule turns into a B. And I'll go into that in more detail later on. You can either then calculate the probability that you converted from an A to B during a time step, or you can sample times like in the Gillespie approach. When a molecule splits apart, so a complex of A, B splits it into an A and B, you have to say, where should I put them? Well, if I put them right at the reaction radius, they'll immediately rebind, right? Because we assume they bind upon collision. So you have to put them in this approach further apart than they bound. So you have to introduce a parameter called an unbinding radius. And one of the things that Stephen did a lot of careful work on was figuring out how to calibrate these binding radii and unbinding radii and other parameters based on kind of well-mixed parameters that you might actually measure experimentally. In MCell, which is another simulator, they don't use unbinding radii. They have some other approach where they try to figure out how much a molecule would have moved to it for a time step to figure out where to place things. And then, of course, if you're simulating things in cells, it's always good to know how to handle boundaries, since most cells are not infinite in size. But basically, you can kind of use standard methods for SDEs here, though there's a lot of work by many people in this audience on kind of coming up with better approaches. But the simplest thing you might think of is if I have a reflecting boundary, suppose I take a diffusion step and end up outside, well, what you can do is just reflect that and then place yourself inside, and that will give you back something that's consistent with the reflecting boundary condition. Okay. So that's kind of one approach for handling reflecting boundaries, and there's other ones for partial absorption boundaries or Dirichlet boundaries. Okay, so there's a number of advantages to this approach. Maybe this is the kind of easiest conceptually of all the methods to kind of actually code up. At a crude level, your time step is decoupled from the density of molecules, so there could be kind of an implicit coupling based on your chemistry and such. But as we'll see, in FPKMC, the density of molecules can really dramatically reduce the time step, whereas here it's a user-defined parameter, essentially. There's several nice public simulators that can handle general systems, complex geometries, crowding, and other features. You can kind of use standard numerical SDE techniques to handle spatially varying drift and diffusion. And the method should be convergent to the underlying models, both the contact reactivity and the volume reactivity. With that said, for the versions that are actually used in practice, I haven't seen any proofs of convergence or derivations of order of accuracy. So I think that's actually a very interesting question. For the specific implementations that people actually use, what really is the order of accuracy? Can we actually show these methods converge? Typically, the way you handle the boundary may not be very accurate in terms of its order of accuracy compared to, say, a PDE discretization of how you would handle the boundary. You need extra parameters versus the RDME approach. You're going to need these reaction radii and unbinding radii. Potentially in the partial absorption model, you'll need rates for partial absorptions. And if you want to accurately resolve biomolecular reactions, you might have to take small time steps, though there is work on how one can kind of modify these methods to take large time steps. Okay? So that's just kind of an overview of some of the trade-offs in using these types of approaches. And of course, this is my perspective. I'm sure if you ask Stephen or someone else, you'll get different answers to what's good and bad about these methods. Okay, what about the FPKMC? How does,
yeah. The last point there. So you said on one hand, the time steps decouple from the density of the molecule. So I guess the issue of these biomolecular reactions is keep having to monitor whether they overlap or not. But some of the density doesn't.
Yeah, so when I say decouple, I mean that you set the time step, so you can kind of set it for accuracy considerations. In FPKMC, the time step is event-driven, so you don't get to pick it. So if things get dense, your time step is just going to be tiny, basically. Whereas here, maybe you can try to do something when things get dense to allow you to take bigger time steps, and of course, that's something that people have worked on. So. Okay. So the second method that I'm going to mention is this FPKMC. So this is the method that generates exact realizations of the underlying trajectories. So the basic idea in the FPKMC method is for every molecule, we put a protective domain around it, disjoint protective domains. Usually we use spheres or circles in 2D, or we use rectangles or cubes in 3D. So I'll use circles for my examples here. And we want to make sure that they're all disjoint. The idea being that within these circles, the molecules are kind of all independent. Until they leave their circle, they can't interact, and their motions are all independent. Okay? And of course, then what we do basically in the algorithm, and I should say, we'll see why in a minute, but your general goal is to try to have the circles be roughly the same size and as large as possible. Okay? Then what we do for every single circle, we basically calculate an event time. So if it's just one molecule, we sample essentially when does this molecule first diffuse out of the circle and where. Okay, so for each of these, I would sample those times. So that's just solving the diffusion equation inside a circle. We all know how to do that, or hopefully we all know how to do that. We can get an analytic solution, and therefore I can exactly sample from that solution to figure out when I would first hit the surface and when I would exit. So for all these circles, I just sample those times. This one's a little trickier because I have a pair of molecules, so I'd have to sample several things. First, when one of them would exit, and then when they might react. Okay? So I have kind of multiple things to sample. But again, I'll explain a little more in a minute. You can still do that and come up with an analytic solution because this is only a two-body problem. Okay? So we can go through and sample all these possible event times, and then the simulation algorithm is just pick the one that's smallest. That's the one that you execute. So if this is the smallest time, I would update that molecule to its exit position. And then I need to now redraw a new protective domain about it. And so I draw one. If the protective domains get too small, I might want to say update the positions of these neighboring ones and try to rebalance the protective domains so they stay the same size. But that's kind of the basic idea of the algorithm, is to kind of march along in each protective domain generating event times and going through in this way. Okay. Yeah?
So, but that sort of assumes that somehow we don't have a lot of molecules around, yeah? Because if you have many molecules around.
So that's what I just said. If, what I was just mentioning with Pete's question is that if you have a lot of molecules, your protective domains are going to get tiny, your exit times will get tiny, and you're effectively taking a tiny time step. So these algorithms are great when you have a dilute system because you basically get away from having to simulate all the diffusion time steps or hops in the RDME. But they're not so great when you have a more dense, crowded system because you're going to have time steps. So really, it's very conditional on the type of problem you're looking at, whether you want to use this or not. So that's correct.
Can I ask a quick question? So you only generate one new exponential, or not exponential, one new random variable for the new circle or sphere, right? You store all the other ones?
Yes, that's correct. And as long as things remain disjoint, there's no problem. Now, if this gets tiny, I could say, okay, maybe I can rebalance these circles in such a way that they're not all tiny. Then I would have to do what's called a no passage update, where I update the positions of these guys conditional on their events not having occurred by that time. Because I know when their events would occur, basically. And you can do that. So then you might have to do a little more work. And you can immediately see what a headache this is going to be, because I'm going to be now having to balance and figure out all these protective domains. And that's why there's only a few people that have actually implemented this algorithm. So, but it's a very nice conceptual algorithm, and there is a public implementation that you can use. And so among other things, it at least gives you a way to verify approximate algorithms. You have an exact algorithm to compare with. Okay, so I wanted to go in a little more detail about how the algorithm works, especially because I don't think I'll come back to it after this first lecture. So let's think about how we do the single molecule protective domains. So I can let x naught be the initial position of the molecule, and the simplest way is to assume it's just at the center of my sphere. D, its diffusion constant. U will be the protective domain, like I said, usually in 2D it'd be a circle or rectangle. Du the boundary. And P of xt, my probability density that my molecule is at position x in the protective domain at time t. So it hasn't exited yet. Then it's just the diffusion equation for how the molecule moves around. When it hits the boundary, it exits. So we have a Dirichlet boundary condition to model the exit. And of course, by picking the simple domain, we can solve this analytically, get some kind of series representation for the solution. So then what we want to sample first is the first exit time. So we can write down the probability your exit time is less than t. It's just given by 1 minus the integral of that solution. There's lots of ways you can sample that, right? A bad method would be to do inverse transform sampling. One way to do it would be to tabulate. That would be a little nicer than just to actually invert this every time. You could try to tabulate this. There's also, I think, in one of the papers by Donev and the FPKMC people, they suggest some rejection sampling approaches. So there's various ways to do it. Once you've got your exit position, you need to sample, once you have your exit time, you need to know where you would exit given the exit time. So you can calculate that by sampling from this density. So here t is fixed, and you would sample for x. So this is a density for the exit position x on the boundary. And again, that's something you can calculate from the PDE solution, though if you start at the center of a sphere, you have symmetry, so it makes life a lot easier. You just pick a random point on the sphere. And then, like I said, you may have to occasionally update the particle's position before it would leave. So if you want to update the position to time t, you do what's called a no passage update, where you sample a position conditional on it not having exited. Okay? And that's the kind of basic way you do single particles. What about pairs? Yeah.
Well, the protected domains are randomly constructed, or there is a rule that you follow to construct?
The goal is to make the protective domains be roughly the same size. That's the general, and then you only encompass one molecule or a pair of molecules that could react at most, and they should be disjoint. So you have those constraints, and then it's basically kind of an optimization problem about how to arrange them. And that's very much the challenge in this algorithm, is figuring out how to balance those protective domains and set them up. Yeah, that's the real hard part. This is the easy part. So, okay. So what about a pair that can react, undergo A plus B, annihilate, say? So if I let X and Y be the positions of the A and B molecules, and these are their diffusion coefficients, use, again, the protective domain, then I have a pair density for the probability density they're at positions X and Y in the protective domain and haven't reacted. Then the pair just satisfies a diffusion equation where each molecule moves in the protective domain. And now I have the Dirichlet boundary conditions that if X hits the boundary or Y hits the boundary, they leave. Or if my molecules hit a separation of a reaction radius, they react, all of which give us Dirichlet boundary conditions. So it's a similar kind of setup. Now you might say, wait, how can I solve this if I assume they're each moving in circles? And generally that's a geometry that we can't write down an analytic solution for. So what you do is you change coordinates. So basically you switch to a separation and center of mass coordinate, which then decouples your equation, which if the particles are in 2D, that would have been a 4-dimensional PDE. If they're in 3D, it would be 6-dimensional. This decouples it into two 2-dimensional or two 3-dimensional equations. And you look at a smaller domain within the protective domain. So you pick that the separation should live in an annulus, right? If you hit this boundary, you react. You'll assume if you hit this boundary, you exit. And again, you assume typically that the center of mass should live inside some circle or sphere in 3D. And you pick R and rho so that this is within your original protective domain. As long as you pick them small enough, this will be a subset of it. Now in this separation coordinates, you have something where I can analytically solve the PDEs because the diffusion equation simplifies in these two cases. And so now you can again convert it into separate equations in these new coordinates. You can solve them. You can sample reaction times versus exit times from them. And then you basically just pick whichever event happens first, whether one of the molecules exits or whether they react. And the details get a little messy, but you can see kind of Donev et al. Talk about this. Takahashi et al., which is the EGFRD method, have how to do this with Robin boundary conditions, which would be partial absorption reactions. They go into much more detail. Okay. So again, I've just told you about a simple case here of how you do essentially diffusion in bimolecular reactions. So I left out first-order reactions. But again, these are typically just internal processes. So each molecule will have a Poisson clock for the time it's going to convert from A to B. So you can just sample from an exponential distribution when it would switch. And that's just another possible event you put in your event queue. The real trick, as I've mentioned a few times, is how to choose the protective regions and rebalance them so they don't get too small. There's lots of, this is very much an open problem, and everything that I know of right now, the codes kind of just use heuristics to kind of try to figure this out. And like I said, it's been extended to the partial absorption case, and in fact, we have a method that extends it if you have drift or non-uniform diffusivity, but you kind of sacrifice your exactness because you can't sample these things exactly anymore when you have spatially varying drift or diffusivities. And the nice thing about this method is it can handle complex geometries. So if you have a piecewise linear or planar domain, then when I'm near the boundary, just make a protective domain that conforms to the boundary, like in this case, a square. I can solve the diffusion equation on a square. If this is Neumann, I still can solve it analytically. If it's Dirichlet, I can still solve it analytically. And so basically, if you switch to boundary conforming protective domains when you're near the boundary, and those domains are simple, then you can actually exactly handle these problems in complex geometries still. So that is one nice feature. And actually, this is something that I haven't seen really been done. I think EGFRD, which is the main simulator for this, just kind of works in simple geometries as far as I know, unless they've updated it recently. Okay, and just to kind of, in the same way I did for BD, mention some of the advantages and disadvantages of the method. So in terms of advantages, one is that this gives us exact realizations of the contact reactivity model, even if you have partial absorption Robin boundary conditions instead of just the Dirichlet boundary condition, and for reversible reactions too. The method works even in complex geometries. It can still give you exact samples. And in dilute systems, it's going to be really nice because you'll take large jumps. You'll avoid simulating all the diffusion like we talked about. Like I said, very difficult to program this. Needs extra parameters versus RDME because you need these reaction radii and partial absorption rates. Determining protective domains is a real pain and something that's a lot of work. And if you have small protective domains, you have a small time step. And then kind of a more general question is, for biological problems, we may have spatially varying drift, say due to interaction potentials or diffusion constants. Is there some way you can extend these kinds of methods to incorporate that? And that I don't know at this time.
So Sam,
Yeah.
when you say exact, you solve these PDEs by some series and then you truncate them. And then you sample. Do you introduce some error at this stage?
So yes, but presumably you can sample them to within machine tolerance if you want. So now whether you practically can, that's a different issue. But I think Donovan and them do a pretty high error tolerance in the truncation of the series. And if you use rejection methods, I think that helps gets around some of the issues that might come up with having to deal with that. So it's exact in that within I think the limits of within the limits of your machine precision, the method in theory is exact. Whether it's exact in EGFRD, the one main implementation, I don't know. I don't know what kind of error tolerance they pick for where they truncate the series. So,
Are there any estimates on computational complexities, things like how many operations it takes to solve a problem with certain parameters?
I mean, there's no estimates that I know of for solving like on 0T specific system. That's highly dependent, right, on the number of molecules and the chemistry and other things. I think Donev has a pretty detailed description of the work kind of per step in his papers. So you can kind of look at that, right?
The question is how many steps are you going to require for any given system?
But that's going to be very dependent on the physics of the underlying problem. Just like with an RDME or a Brownian dynamics simulation, how many steps you take is going to depend on how well you want to resolve it, right? So. Okay, any other questions? Yeah.
How do you handle the dissociation step?
How do you handle the dissociation step? So I think the way that it's done there's many ways one could conceivably try to do it. You could try to solve the pair problem and do that. I think, though, what's typically done is it's decoupled from association, and it's just done as a first-order reaction. So things associate, and then they get an exponential clock to dissociate.
But then where do you put the molecule? You put it in the
That depends on what you want to do. So actually, this is something I was going to talk about in more detail later on. But so one method is if you're using a volume reactivity model, you could just put things where things dissociate because they don't immediately necessarily rebind. If you're using things react upon collision, then you would typically place an unbinding radius away and sample uniformly on the sphere. If you're using partial absorption, then you would just place essentially right by the boundary surface, right? Uniformly across the boundary surface.
Then you could get immediate reabsorption, couldn't you?
It could. Well, it wouldn't be immediate, but it could be very quick, right? I mean, it could be very conceivable that you rebind quickly. That's right. So yeah. So there's different options, and actually, what you should do, what the best thing to do, I think, depends on a number of conditions. But yeah, so that's something I'd be happy to chat about more if you have thoughts. So, okay. So the last thing I wanted to talk about was the RDME. So I have till what? I have 25 more minutes. Yeah. Okay. So the last model that I was going to mention was the RDME. So in the RDME, basically, we keep track of the total number of molecules of each chemical species in each lattice. That's what I had said earlier. And again, the molecules hop around on the lattice. So here I have 1A in location I, 2B. Here in location I plus 1 minus 1, I have 1A, 1B, and so on. And then every molecule has just rates that it can hop from its lattice site to the neighbors, which on a Cartesian grid like this would just be dA over h squared. And again, as we saw earlier, these are just going to come from discretizations of the Laplacian. So the goal is basically, if you wanted to use unstructured meshes, to cook up discretizations that you can interpret as generators of Markov processes that describe continuous time random walks with certain rates. You can think of this as just the first order reaction. So in particular, if I group molecules in the same location together, then the rate for some molecule of species A to hop from here to here is now 2 dA over h squared, right? Because I have 2 molecules here, so the rate doubles. So I just kind of think of this as like a reaction, that AI goes to AI plus 1, 0, the neighbor, with some rate dA over h squared, or I should say propensity. And so as we saw yesterday, then we basically can just simulate this all with the Gillespie method, right? At every site, I would have my bimolecular reaction with some rate k. I'd have my dissociation reaction with some rate, and then I have the hopping to the 4 neighbors, and then I'd have all these reactions possible at every site. So I just have a giant list of reactions, and I can throw this into my Gillespie method or first reaction or next reaction method and simulate it. Okay. Now, of course, there's optimizations you can think about doing, and hopefully I'll have a little time to describe them tomorrow, but that's kind of at a high level how it works. Okay, so there's lots of things I've left. Yeah.
You should use, an exact method. It will not, computationally very costly to use the Gillespie algorithm or the method. Cannot use an approximate method and try to control the error.
Okay, so you're talking about for solving the RDME.
Yeah.
So sure, you can use approximate methods. You can do tau leaping, like we heard about yesterday there. You can do, you can do analogous things to chemical Langevin equation. And people certainly do that, and they try to couple, you try to have systems where when you have higher populations, you kind of resolve things with tau leaping and lower levels with Gillespie. But for the purposes of this one, I'm just going to focus on the basic exact simulation method. I probably won't have time to go into kind of coarser grained methods, but you're certainly right. And actually, an open problem, as far as I know, is adapting these multi-level Monte Carlo methods that we heard about yesterday, and I guess we'll hear about many more times, to these spatial problems, where there's some tricks, there some difficulties in getting things to couple well come up. So you'd like to have some strategy like that available to reduce the number of simulations you have to run. But I don't know, maybe someone who's speaking later in the week will tell us how to do it. But otherwise, I think that's an open problem as far as I'm aware. Okay, so there's lots of extensions. So one of the reasons that people like the RDME is that since the diffusive or the spatial transport looks like it's just discretizations of the continuous models, we can use PDE discretization methods. So we can apply adaptive mesh refinement if we want to kind of refine the grid and get higher resolution. We can add in advection or drift due to a potential. There's GPU versions. There's lots of other extensions people have done. As he just asked about, you can couple up to things like tau leaping or reaction diffusion PDEs. And we can handle complex geometries just using standard PDE discretization tools. So we have methods using finite volume approaches where you cut your voxels by the boundary and you modify your hopping rates to account for it. And these just correspond to certain types of finite volume discretizations with cut cells. Some of the groups in Uppsala, a number of people who are here from the group, have done unstructured mesh methods based on finite element approaches, so where you can hop around on unstructured meshes and still get things that converge back to the underlying diffusion process. So there's lots of kind of nice extensions to the RDME to facilitate biological modeling and features that we might want to have. There's a lot of publicly available simulators, as I mentioned earlier, like STEPS and PyRDME. And the RDME generally requires less parameters than the other methods. You don't need to do this kind of parameter calibration of reaction radii and things that are not generally measured experimentally. Typically, you just have your diffusion constants and your well-mixed reaction rates, and then the only other parameter is the lattice spacing. Okay. Unfortunately, that's a big, important parameter that's very difficult to deal with. Unfortunately, the drawback to the RDME approach is that the bimolecular reactions are very lattice dependent, how they get modeled, because you only allow 2 molecules to react when they're at the same lattice site. If you take your lattice to 0, you get point particles moving by diffusion. If you're in 2 or more dimensions, particles moving by diffusion can't find each other. So you end up losing bimolecular reactions if you take the lattice to 0 in the RDME. So what we'll see hopefully later on is that the RDME, if you don't have bimolecular reactions, is just a discretization of the continuous particle models. But if you put in bimolecular reactions, then it doesn't converge back to them. Now, you can find ranges of lattice spacings where the RDME may be a good approximation to the continuous models, or where you're not losing bimolecular reactions, but it's a little tricky, because you can't take your mesh too big. If you take it too big, you approximate diffusion inaccurately. If you take it too small, you're going to start losing biomolecular reactions. So you need to kind of find a sweet spot where it's neither too big nor too small. And unfortunately, that often tends to be very dependent on the specific chemistry of the system you're looking at, how many molecules there are, what the chemistry is. So that's a little tricky. And there's a lot of work getting around this. Linda's group has a number of methods for kind of tweaking how you do biomolecular reactions, the effective reaction rates you use to give you a much broader regime of lattice spacings where the RDME approximates the spatially continuous models well. But if you want to get something that's convergent, we've proposed a method based basically on discretization of the continuous volume reactivity model that gives you something that looks like an RDME. It just adds the slight difference that now, instead of only reacting with molecules at the same site, you can react with molecules in neighboring sites. And then this basically allows you to have a method that converges as you shrink the lattice down. And then the other big challenge with the RDME is you spend a lot of time simulating molecules hopping back and forth. So these molecules hop with rates d over h squared. If you have a very fine lattice, those rates are really big, and you generally just spend 90% of your simulation time simulating molecules hopping. So a lot of the other work that people have done has been on trying to come up with ways to speed up or approximate the kind of hopping so you don't have to simulate every single one. Okay, so that's kind of a high-level overview of the different numerical methods. So I'd kind of like to end this kind of first piece by just saying a little bit, showing you one example of a biological system that these models have been applied to, just so you can see what one of these simulations looks like. So this is actually from 2012, so probably they have even more impressive stuff. This is from the M-cell group, and what I'm going to show you is a simulation of action potential initiation during synaptic release. And in particular, we'll see the action potential come in, the vesicles will dump their neurotransmitter, and then we'll start seeing kind of signaling here in the postsynaptic region in the depolarization of the spine. So actually, this is not just simulating the particles moving around, reacting, and diffusing, but it's actually also doing simulations of the propagation of the action potentials across the membranes. And this is courtesy of Thomas Bartle. And when he gave this to me a number of years ago, he said, please point out that this is the work of an enormous number of people, from getting the data to actually getting the meshes out of the imaging data, and then the kind of underlying kinetics models, and then actually doing the simulations. Okay, so let me show you that, if I can find it on my computer, because I forgot to pull it up. Documents, presentations, movies. And it's none of those. Ah, here it is.
And just since I don't know if all the students know what action potential is, do you mean the
right.
chemistry?
Right. So you have a transmembrane potential difference between the inside and the outside of the cell, which is usually negative. And in neurons, if you stimulate sufficiently, you can cause ion channels to open in the membrane of the neuron that allows this a kind of wave of electrical activity to propagate across the neuron. So that is, the potential will shoot up. That's the initiation of the action potential, and kind of propagate across the cell. So you get kind of the potential goes up, and then it goes down and recovers, and this moves in a wave through the cell. And those action potentials moving along an axon will cause the synaptic vesicles in the presynaptic region to dump their neurotransmitter. And that's what we're going to see in the movie. Okay. So hopefully that's clear. I'm happy to talk in more detail if anyone hasn't seen that before. So this is a little chunk of an EM reconstruction. This is one neuron in gold. These are astrocytes. And then this is, I think, the kind of terminus of an axon from another neuron here in green. And we're going to zoom in if this starts playing. Ah, okay. And the time scale, I guess, is logarithmic, so keep that in mind. And you can tell that Tom had a good time making this movie, because it's a very fancy movie. Someday I will make a movie as good as this, and then I'll be really happy. So you can see, I'm not going to even try to explain all the chemistry that's going on here. Instead, let's just kind of use it to get a sense of scale. But there's thousands, I think tens of thousands of molecules that they're simulating here. And these were from 3 years ago, and I'm pretty sure that this was kind of run on a supercomputer for these types of simulations to actually go through. So you can see all the little molecules. And we're going to zoom in here. So here's the presynaptic region, and if you watch the color, I believe brighter is higher potential. So it's what's getting activated. I believe these little yellow guys are calcium. And eventually we're going to get the synaptic vesicles will all kind of start releasing. I don't remember what the synapse here is, but you can see that the neurotransmitters that get released diffuse outside the cell. Some of them bind to receptors on the surface of the postsynaptic region in this spine. Some of them get soaked up by the astrocytes and removed from the region. And you'll see now we're going to kind of start getting activity, bursts of activity now in the spine in response to the action potential, the transmitter that came across. Okay, so maybe I'll just let this run. So it looks very fancy. I don't know what's going on actually though here in terms of the chemistry. I mean, there's like 30 different chemical species that are in here, and I'm not sure exactly. All I know is that the little yellow ones are calcium. so you can see it kind of get these bursts of calcium appearing. But I think that gives you at least a sense of scale of what people can do with these kinds of models these days, kind of what you can get up to. So I guess here we had a second action potential kind of initiate and go through.
So, Sam,
Yeah.
if I'm allowed to, I would like to say that actually this is very nice, but there is a lot of science fiction in all of these movies. For example, we still do not understand the process of how a vesicle is released.
Uh-huh.
So all of this piece of information are actually, hidden, or they invent a way to activate a vesicle.
Sure.
So I mean, this has to be said because this is not the answer. This is just one.
Of course. Well, no model is the answer,
was-
right? Every model is a cartoon of what's going on. So if you have to look at you have to go to the actual papers and look at the predictions they make with it, and they work with experimentalists, so you can see how well their predictions guided or gave insight into the experiments to assess how reasonable their model is. But I don't know the details of their model. I'm certainly willing to believe that there's a lot of mechanisms that are just kind of put in here to make things work the right way. That it's not all well-known, well-established interactions. I certainly believe that. So, I mean, for anything with this level of detail, right, there's going to be components probably that you don't know exactly how they work. So, but that is a good point. So, okay. So that was kind of everything I had for my opening lecture, which I thought would be a lot shorter. So I also thought I would be giving this later, so I had see you in 30 minutes after your coffee break. Though I guess we would be done with the coffee break probably at this point if I actually would. So hopefully I will maybe see some of you tomorrow. Because I have...
You have until 10:35.
I have till 10:35. Okay, well then maybe I should start a little bit of what I was going to talk about tomorrow. And then we can break at that point. Okay, are there any questions on this kind of opening? So this was kind of the overview. Now I'm going to kind of zoom in on some pieces of it in more detail. So, yeah.
So for the reaction diffusion master equation, as you described it, do you see lattice artifacts where you observe diffusion happening faster in directions parallel to the coordinate axes and slower off, on the diagonal, etc.? I mean, because it seemed like you would observe that.
So only, to the same extent that you would when you solve the diffusion equation on a Cartesian grid, right? If your grid is not refined enough, you may have artifacts due to having too coarse of a grid. But ignoring bimolecular reaction, if you make your, then it should converge to the continuous models, and if you make your grid fine enough, it should be able to provide you with the level of accuracy you want. So I haven't, in any of the models I've looked at, seen anything that indicates to me that there's artifacts. I certainly believe that if you have a coarse grid, right, and if you have something where you have very anisotropic diffusivity or something, that may not be captured well with a Cartesian grid. But I haven't looked at any examples where that occurs. Yeah, I don't know. Did you have a specific one that you know of or that you've seen?
No, it just seems like that from the description that ought to happen, but I maybe just need to think about it some more.
Well, the question is, are you willing to use the discrete Laplacian in a Cartesian grid to solve the diffusion equation? Do you believe that you're going to get artifacts there? Because that's basically all you're doing here in some sense. So, yeah. Okay. So I guess my plan at this point was I was going to kind of go through in more detail how you kind of derive the Brownian dynamics methods. So far, I kind of just focused in on kind of at a high level explaining to you how they worked, and I thought so I would show here how you actually can write them down mathematically and how everything kind of arises from the same kind of general formulation. And like I said, it can be a little notationally messy, but hopefully it kind of gets clearer as I go. So why don't we maybe get that established to start, and then we can tomorrow go into more detail about how the numerical methods come out from that. Let's see. And I'm going to switch to the blackboard, I think, from now on. So it's chalk. Is there chalk? Okay. So let's see. So I gave you a lot of notation, and I'm going to start using it now. So please feel free, if the notation at any point is confusing, to ask questions or ask me what something means, since I'm kind of assuming we've all absorbed it. So what I want to kind of build up to is an example that lets us see. Does this one move too? Yes. Okay. An example that lets us see kind of just a general set of reactions that would cover maybe all the cases we look at. So we want to kind of build up to. And is that big enough for everyone to see? Yes? Okay. So I want to build up to a system of this kind of form, where we have a zero-order reaction, a second-order reaction, and then an association reaction, and a dissociation reaction. So kind of basic set of elementary reactions. And so the idea is to kind of write out the mathematical model for the volume and contact reactivity models to see what they look like for this general system, and then see how probably there's no way we'll get to this today. Tomorrow, to see how if we do operator splitting, we get Brownian dynamics, okay, and explain how we would then simulate it by Brownian dynamics in more detail. And then also in the next lecture after that, explain how basically we could then get an RDME or a convergent RDME all by discretizing the same equations. And basically, the operator splitting is like doing a time discretization. The RDME is going to be doing a spatial discretization. And then it just comes down to doing that discretization in the right way to get something that you could think of as representing a Markov process. So, okay, and let me see. So there is going to be one little issue. So we're going to assume we work in a bounded domain. Call it omega. Omega is going to be my domain. And I think at some point I'll need it. So eta of x will be the outward normal at x. Okay. My eta doesn't look like an eta. And is there any other notation we're going to need on the domain for now? Ah, right. So omega is the domain, and the boundary will be d omega. Okay. So just a little bit of notation. And that's the kind of setup for what we have. So let's kind of start going through kind of hierarchically how we can write out the model for these different cases, adding them in one by one as we go. And I don't know if we'll get through all of them today, but hopefully we will have time and finish them up next lecture before we do the simulations on them. So of course, the first component is the diffusion. So just like I had before, right, my state is going to be given by the number of particles of each species, right? So I have a stochastic process for the number of A, the number of B, and the number of C. Little a, little b, little c will be the numbers. I'm not writing everything again because I had it in the previous talk, but again, ask questions if it's not clear. And they'll have position vectors QA, QB, and QC. Okay? And remember, QA has its components are the positions of each A molecule, right? So if you think about it, right, this guy is in omega to the what? You have A copies of omega. This is in omega to the B. This is in omega to the C, or I mean the product of that. Okay? So first, diffusion. Diffusion's the easiest thing. Okay. So if you remember, we had these densities. Actually, I guess I should write the density. That's kind of key. So our density is FABC of QA, QB, QCT, which is my probability density, right, that we have state QA, QB, QC at time t. And I guess before I write the diffusion, the one thing that we need to mention is how do we normalize this? So if you think about this, my state variables are the numbers of each species, the position of molecules. So I want to normalize by integrating over positions and summing over the number of molecules. Okay? And there's the technical point that I had before about the symmetry in the arguments. So we're going to have a slightly maybe unusual normalization arising because of that. Okay, so to normalize this, the first kind of step, man, there's a ton of these. The first step is to just look at what the integrals are. So I'm going to call the probability that A of t is A, B of t equals B, and C of t equals C. Let's define that to be given by P of A, B, C, T. Okay, so this is essentially would be the density for the system ignoring molecule positions. Okay, so what we would probably naively think is it's just the integral over all QA, QB, QCs of this. But because I assumed molecules are indistinguishable and that I'm basically going to think of this as a symmetrized density, like I said, you can flip two components of it, we're going to get a slightly different normalization. So the normalization we're going to get from our system is going to be that P of A, B, C, T is going to be given by, let me make sure I get this right. So the integral over omega to the, I guess, A plus B plus C, one copy of omega for each molecule, of F, A, B, C of QA, QB, QC, T, dQA over A factorial, dQB over B factorial, dQC over C factorial. So the factorials are so that we don't overcount a given point because we've symmetrized things. Is that okay with everybody? So if I hadn't symmetrized, then I wouldn't have to have these, but then my molecules would be distinguishable. Yeah?
Do you lose some information by doing that, or?
Well, yeah, so I can't identify a particular A molecule, right? So I can't distinguish between them anymore.
But is there a consequence for like?
There's no consequence for anything I'm thinking about. I mean, if you want to tag a particular A molecule, right, you can't do that in this formulation anymore the way I've done it because they're interchangeable. You have to go back to the unsymmetrized version. So that's right. Okay, so this will be useful later because what we'll see later is that as long as we don't have second order reactions, if we have systems with just zero or first order reactions, these satisfy the chemical master equation that Dave talked about yesterday. So they satisfy the well-mixed model. Okay? So it's only when we add in bimolecular reactions that it stops satisfying the well-mixed model. Okay, so then my normalization is that now the sum over A going from 0 to infinity, and B going from 0 to infinity, and C going from 0 to infinity of P of A, B, C, T is 1. I have to have some state. I have to be in some state. Okay? Is that okay with everyone? So that's going to be our basic assumption on the state of the system. So, okay. So this is important just for when we calculate statistics. We need to have this kind of setup. So, all right, good. So that's the first bit of notation. So why don't we look at diffusion, and if we have time, zero order reactions, and then that's probably a good place to stop for today. So diffusion so I'm going to have an operator of the following form. I have that the time derivative of f of a, b, c of qa, qb, qc, t is going to be equal to some diffusion operator. And for each a, b, c, I'll get a different diffusion operator acting on f. OK. I told you the notation is horrendous here. So like I said, if someone knows a better way to do this or can think of one, please let me know. I would love to use it. So this is the diffusion operator for when I'm in the state where I have A molecules of species A and B and C. And what's that operator? Well, it's just continuous Laplacians in the various coordinates. Oops, little b. dc Laplacian qc. Does that make sense to everybody? So I just take Laplacians in every individual coordinate, and for each species they have the appropriate diffusion coefficient. So basically this is just a kind of fancy way for a multi-particle system of writing down the operator that gives me Brownian motion, corresponds to Brownian motion of all the molecules. Okay, so that's the first step. That's diffusion. What about a first order reaction? So here that's a little more interesting. So let's look at our 0 goes to A reaction. And in that case, we need a little bit more notation. So I'm going to let the magnitude of omega, let's just write it like this way. Let's let V equal the magnitude of omega, which is the volume of the domain. And to be honest, nothing I'm doing requires us to be in 3D. So if you're in 2D, it'd be the area of the domain and so on. OK, so that's V. And let's see. So now we add in another operator that corresponds to the 0 order reaction. So this is my, so we add an operator, call it R0 A, B, C, OK, for the 0 order reaction. So our game plan here is for each reaction, we're going to add in another linear operator to give us our final model. And so basically, we've got to write down 4 different linear operators for the 4 reactions we have up there. So what is R0? So let me give it to you now, and then you can kind of think about it overnight. R0 acting on F is the following. It's alpha, the sum from I equals 1 to A. And how do you get into the state where you have A, B, C? Well, you have to have 1 less A. And you have to throw away one of the A's. So that's how I come into the current state. I'm in a state where there's 1 less A. I throw away the ith one. So I have to sum over all of them because I throw away each possible one. Each one of those brings me in. And alpha gives me the rate I come in. And I guess I should have said I'm going to assume that the rate alpha is the rate of the reaction. And the units I'm going to assume is it has units of, let's see, what is it? 1 over time times volume, right? So if you want to get a rate, you have to multiply by volume. But that works out fine here because here this has 1 less unit of per volume. This gives you the extra one so that you get the same units as F, A, B, C. Okay? How do you leave the state? Well, there's just a rate that you lose a molecule, which is minus alpha times the volume times F, A, B, C. So that's it. That's my operator. I have the incoming flux term and my outgoing flux term. And leave it for you to think about that and see if you can convince yourself that it makes sense to you. So let me give you one exercise to try tonight, if you want, or if this is of interest. Show the following. Show that for the model I've given you so far, where you have just diffusion and the 0th order reaction, that the following is true. It's the following what? You have alpha v p of a minus 1 b c t minus alpha v p of a b c t. And I gave you the definition of the probability you have a given number. So this is just the well-mixed chemical master equation. So if you want to make sure you really, the notation and the kind of setup and the normalizations make sense to you, show that the particle model, if you integrate out all the spatial coordinates, just agrees with the chemical master equation for this simple case. Okay. Yeah?
There's a notation question. I mean, the way that you have the operators written, like R acting on F of A, B, C, I mean, it's not, how shall I say this, A, B, and C, that's, you don't really mean that it's just operating value-wise on A, B, and C, right? For example, your R acting on F, A, B, C refers to F of A minus 1, B, C, right?
R acting.
I'm just saying that equation you just wrote down, R naught of A, B, C acting on F of A, B, C.
Right. So it pulls in other values, right? So it's like it involves shifts in the state. That's right. So the way we're going to ultimately formulate this, think of this as we're writing down the components, is we're going to formulate a vector of the F's and a essentially matrix or an operator of the operators that will give us the full system, which is called the second quantization representation by Doi. So I'm basically going to the Doi representation.
But for now, I guess, can we think of it like R operates in F and then you're evaluating it at A, B, C?
Sure. Yeah. Yeah. You could think of it that way if you want. That would be perfectly fine. So, the only reason I'm putting this superscript in is I'm going to use without the superscript to mean the full operator later on that acts on the state vector with all the F's. So, yep. Okay. Any other questions? Yeah.
So for the reaction that leaves the state, why do you scale with V and not with A?
Right, because if I'm in a and not with what?
Little A.
Little a?
Is that the number of A?
Because it's a 0 order reaction, right? So the rate that an A gets made doesn't depend at all on how many there are. It just depends on the size of the domain, right? So if I double the size of the domain, the overall rate will double that I'll make molecules. So alpha V is the overall probability per time that a molecule gets introduced, and I'm just assuming it's placed uniformly. That's why there's no spatial thing here. It just gets placed uniformly.
But if it would be the death process?
You mean like A goes to 0?
Yeah.
Then, yes. And so tomorrow we'll write out the other 3. A goes to B will have an A in it, just like you said. A plus B goes to C is where we start seeing the differences in the models that we're thinking about. You know, we're volume versus contact reactivity models. And that's where things kind of start getting a little tricky on how to write down all the terms. You're right. So the main reason I'm writing it this way is that once we have the equations formulated in this way, it immediately becomes clear how Brownian dynamics arises from them, essentially. So that's the point of this. And the RDME, actually. So, okay. So I'll stop there because I think now I'm a little over. Is Johan Paulsen going to give his talk? I guess we have coffee break now?
So, yeah. We're going to break until, let's say 11. We're going to check in on Paulsen, and hopefully he'll be well enough, and we'll continue with the talks at 11.